I'm delighted to introduce our next speaker for the session, Dr. Ioannidis. Alex is a faculty in genetics and biomedical data science, and he's previously trained at Harvard and Cambridge, and we're very happy that we have him here at Stanford. Alex does a lot of extensive research on building new computational tools for the analysis of genomic data, structured data, and we're excited to have Alex join us and share some of his work today. So Alex.
Great. All right, so I get to talk to you today about genomics and about precision medicine and predicting disease phenotypes. OK, so let me, sorry, do I advance? There it is. All right, so a quick introduction. All right, so we're going to go back into computation now. I'm going to take you from the world of computation into the world of health and medicine. So my background was very computational. At one point, I was even doing superconducting computing, as up here, and quantum computing. And I have moved very heavily into deep learning, trying to design new methods, really, and machine learning. So here's a recent hypernetwork paper that we have that we encourage people to use. We think it's the best out there. Hypernetworks, by the way, for those who don't know. So the traditional machine learning model, right? You're going to try to pick your model of choice. You're going to train hyperparameters. You're going to cycle through. So it could be a random forest, could be XGBoost, something like that. You'll try this many times, and then you'll have something that works with your data, and you'll deploy it. So a hypernetwork actually generates the weights that will work across a wide variety of domains. So you just put your labeled data in, you run, in this case, our network hyperfast, and then you get your model out. So the model has been trained on a wide variety of data sets of different sizes, of different modalities, of different types, all of this. In this case, this is tuned for tabular data, which is very, very common across medical and biomedical domains. Think of large tables, right? And even genomic data is tabular data. We're going to get to that in a minute. So we have really nice performance. You always do really great on your own benchmarks, right? But here we show great accuracy across a wide variety of data sets. Our method here in blue. The others, you may recognize some of these like GBM and XGBoost, et cetera, not doing as well. And we're ranking near the top down here. Here's our rank. And this is a network, though, that you don't have to retrain. You don't have to retune, really. You just, out of the box, run it on your data. So this is what, this is really important, of course. We're hearing a lot about biomedical data sets, and of course, the methods that you're actually using are being rapidly developed. And I encourage people to contact us if they're interested in trying out. We have a number of methods like this. Again, hyperfast works extremely well. Variety of data sets here. I'm showing you diabetes phenotype prediction, so predicting people who have cases who have diabetes, who don't, based on biomarkers, et cetera, and a genomic data set. And we have another method we're working on now, an even large tabular model, ILTM. So anyone interested in tabular data, please talk to me. But what is the most really exciting data sets to me right now in biomedicine? I'm very excited about genomic data. Now, genomes have been around for a while, but what we have now are really, really large databases of genomes. So we're talking about hundreds of thousands of individuals, complete with their medical records, whole genome sequence. And why are genomes interesting? Because think about how many features you have in a genome, right? Millions of variants that are different from each individual to each other individual, right? So incredible number of features, but very, very well typed, right? So you can think about the noise that you have in lots of other data sets. In genomes, you have now, with high quality, high coverage genomic sequencing, you have extreme precision for all of these variants. So you have lots of features which we know are very important, right? In predicting phenotype and disease, but we're a long way away from being able to do it extremely accuracy, so it's an exciting field of research. If anyone who's seen identical twins knows that there's certainly something in the genome that does express itself in physical traits and disease risk. And we're not able to predict that as well as we'd like to. Computational models are what I'm focusing on for that. So here's a model that we published a little while ago, just illustrating large data sets of genomes, so these large biobanks. Here you see it run on All of Us, which is a really exciting database of Americans. We have hundreds of thousands of individuals, whole genomes, all sorts of phenotypes and disease traits for these people. And here you see the diversity of all of us. So this method lets us see the clusters based on genetic ancestry. And so the colors, every single bar here is a different person. You see these very narrow vertical bars. The data set is huge, as I said, hundreds of thousands of people. So you're visualizing all of them all at once. And the color along the vertical of each bar tells you what percent that particular individual derives genetic ancestry from the appropriate colored label here. So you have African ancestry, European, East Asian, et cetera. And so you just see actually a lot of variation. And here we can see for every chromosome, actually, of every individual, I've broken one tiny column individual out here, and one of their chromosomes, we can see two haplotypes, right, paternal and maternal. And we can see the different blocks of their genome, which are coming from different ancestries, because many of us in the US are coming from a variety of ancestries. In fact, the majority of children born in California now have multiple ancestries. So very important problems, and I'm going to be talking more about this in a minute. But first, I'm going to walk you through a story of computational genomics. This just got published a week ago from our group. And so basically, what I want to do now is take you through two concrete stories for the rest of this little discussion. So I'm going to talk to you about this story and show you some of the things we can do computationally with genomes. And I'm going to talk to you about another story talking about precision health and genomes in the context of global health. OK, so what do we do here? What we do is a variety of things. So we show how you can do really, really good compression of genomes using this method that we introduce. Why does compression matter? Well, as again, we've got genomes, whole genomes have millions and millions of variants. We've got hundreds of thousands of them now. It's growing rapidly. Each country's doing its own biobank. We've got biobanks in the Middle East. We've got biobanks in Asia. And compression is an enormous problem. If anyone's worked with whole genomes in biobank data sets, they can tell you that the data is getting very unwieldy. So that's one thing we can do. And I'm just trying to give you sort of an overview with the time I have. Another thing we can do, again, looking just at computationally at these genomes, is we can analyze entropy, which is a measure basically of genetic diversity, right? It's something that comes up when you're thinking about compression methods. But it also comes up when you're talking about actually variation in the genome, right? Information content, the amount of disorder or entropy in the genome. And you see that there's more diversity, as we would expect, of course, in the region that humans emerged from, right? So all humans come from Africa, and there's much more diversity there. And I want to tie this back to something that Barbara mentioned, which is that the current data sets, and I showed you a picture of all of us, are really lacking in diversity. All of us is probably the best we have today, but we really need more. And it's not just a question of underrepresenting most of the world's population, which is true. That's happening. We really oversample European descent genomes. But it's also about representing genetic diversity, right? If everyone came out of Africa ultimately, and there's more genetic diversity in Africa than anywhere else in the world, then we would probably want to sequence a lot of individuals in the region of Africa where there's so much diversity, right? Especially if we're trying to understand what different variants do in the genome, right? We want to see as many of those as we can. And you see that here. The most entropy is in African genomes. OK, so great. And we can do visualizations with this method. And that's very important and interesting to visualize diversity, et cetera. OK, so now I would tell you about the other story with the 5 minutes I have left. So this is a preview, sort of. This is the cover that you haven't seen yet, but now you have. This will be the cover of Nature Medicine for February. So our article made the cover, and here it is. It's the article you can read already because it's out early access online. And we're going to walk through clinical genetic variation in Hispanic populations in the Mexican biobank. So Hispanic populations, everyone has this sense of this category, Hispanic, right? And we would like to tailor things in precision medicine much more narrowly than just some broad category like that. And why does that matter? So in this article, we're trying to, can I go back? Yes. We're trying to go into a little bit of depth of why that matters. So here we're looking at a variant in CYP3A4, so that actually affects metabolism of fentanyl, amongst other drugs. And you can see a really large frequency difference within Mexico, right? So you see frequencies down here of over 0.6, and up in the north you see much lower, okay? So that's just within Mexico. Of course, there's a lot of people with ancestry from this region in the United States. This is very relevant to patients in the US as well, right? And you can imagine what the variation might look like in the rest of Latin America, right? So to just label an individual as Hispanic and then try to tailor personalized treatment based on that, you can see you're going to have some problems here already. Now, looking here, many other variants that we dive into in this paper. We're just looking at a map of this one. We kind of deconvolve it into two components. We look just at the pieces of the chromosomes in all of these Mexicans that are European. I showed you how we can look at the different pieces of the chromosomes. And you see that the frequency in those pieces is low, and the frequency is high in the indigenous segments. So you might say, OK, so this is a result of the difference that you see in indigenous American ancestry versus European ancestry, which are both present in Mexico. So maybe it's as simple as that. Well, for this variant, maybe. And by the way, I want to call attention to this because you're going to get a really great talk on why these variants matter, I think, next on pharmacogenomic variants from Terry. But we can go a little deeper for another variant. And this variant, we're looking now at its frequency in Mexico, looking only at segments that are indigenous American in ancestry. So now we're controlling for the different large-scale differences that you see. Some people have pieces of their genome that are coming from Europe, from Spain, usually Mexico, and some people have pieces that are coming from indigenous American background. OK, but if we look only at the people who have segments coming from an indigenous American background in this gene, what we see is still a large variation geographically. In fact, very high frequencies in the Yucatan, and much less in parts of northern Mexico. So in fact, the variation in frequency that we see across the states of Mexico in just the indigenous ancestry here are larger, and that's what we're showing down here, than the difference you see across continental populations. So populations like groups from East Asia versus groups from Europe. So there's more variation in Mexico in just the Native American genetic component than there is across worldwide populations. And you say, why is that? That seems odd. Well, because you have these founder effects. You have small groups of individuals. Remember, the Americas were settled by small groups that came across the Bering Strait, so you already have a very strong founder effect, which creates pronounced genetic drift. And that continues as the Americas are settled through these narrow isthmuses, through Central America, et cetera. And so you really have these strong effects that persist to today. And this matters for precision medicine. And I love this quote from William Faulkner, which is, he said, the past is never dead, it's not even past. And that's true in our genomes, and it's true then for precision medicine, because that is based on our genomes. And the last thing I want to show you is another variant, just to tie it back again to why all this is so important. So this is a variant in the biotinidase gene, which causes deficiency in recycling of biotin, right? Which is a really big problem. It creates all sorts of neurological deficiencies, but it's very easily treated by biotin supplementation. And what do we see? Well, it's not really a problem. It's at very low frequency in most of Mexico, but in the Mayan ancestry region, in the Yucatan, it is actually present. And this is important to know. And it's not just important to know if you're a clinician in Mexico, which is a large country, very important, but in the US as well, because we have people here, lots of people who derive their ancestry from Mexico, who we may be giving a single label to as Hispanic, but should really be getting different sort of treatment and genetic testing depending on where in Mexico they're from, where their ancestry comes from. And so this is something that we really want to highlight in this paper, is diving deeper into these backgrounds. And it's not just true, of course, for Mexico. This is true for the whole world. And really exciting that we're in a place now where we can start to do this for other parts of the world as these large biobanks of whole genomes come online. So I'm really excited about all this. The paper goes through lots and lots of variants, and you can see up here, and there's a MEXFAR database that can be accessed, and anyone who has patients with ancestry from this region can look up now online and see what they should be thinking about more deeply depending on the ancestry and location of origin of those patients. And finally, I really want to emphasize this is all a collaboration with a close collaborator of mine, Dr. Andres Moreno Estrada, who runs the Mexican Biobank based in Mexico City. And here we are returning some results to a participant. It's very important to include the participants who have volunteered, right, their own genomes to be part of this, to include them in the results. And we did that. Actually, this is a picture of the chromosomes of this individual and the ancestry. You saw one of those pictures yourself earlier. And here's the student who led the work here this fall. And this is the student who did the really great work in the genome research article I mentioned on compression of large genomic data sets. So thank you.